This section presents learning objectives 9 through 12, covering bite marks, blood analysis, firearms, and question documents. The video offers a brief introduction. Professor Reese advises reviewing the material thoroughly for a full understanding of physical evidence and forensic science in investigations. Most bite marks usually involve humans and are easily distinguished from those inflicted by animals. Victims may be alive or dead. An avulsed bite is so forcible that a portion of the body is detached. This may be a finger, a chunk out of someone's arm, or some other portion of the body. The collection of bite mark evidence from victims, when reasonably possible, should be left to a forensic dentist because bite marks may be mistaken for bruises, abrasions, indentions, or lacerations. Chyloscopy uses the unique wrinkle patterns of lips to identify individuals. Ear marks can help compare and potentially exclude or include someone as a suspect. Dental profiling, used when other identification methods are unavailable, allows forensic dentists, sometimes working with anthropologists, to assess a person's age, gender, race group, habits, and social class based on teeth and skull condition. Hair evidence is common at crime scenes, as victims and suspects can easily transfer it through contact. Hair can be found on ski masks, clothing, hats, wigs, and in the hands or under the nails of victims, making it straightforward to recover and analyze. The following are some things the forensic examination of hair can reveal: whether the hair has a human or animal origin, racial traits and body area source from which the hair was removed, method of removal, for example, was the hair forcibly removed, evidence of damage such as decomposition, trauma, or burns, drug use and timing. Are contaminants such as blood, semen, soil, pet hair, and paint present? Are hair treatments such as bleaching, dyes, and shampoo present? Possible disease indicators. Suitability for microscopic or DNA analysis. Comparison of known versus unknown samples. When collecting hair standards, at least 30 samples should be obtained from different head regions, front, back, top, and each side, to account for all variations in texture, color, and length. At each site, half the hairs are pulled, and the remainder is combed lightly onto clean paper. Use gloves and a clean comb for each body area. All relevant areas should be thoroughly combed to remove foreign hairs and materials. Hair samples can be stored in a sealed labeled envelope, pillbox, or other airtight container, as accepted by laboratory protocols. Known and unknown samples must be packaged separately before submission to the laboratory. The investigator should be present when qualified medical personnel collect samples and confirm that collection and packaging procedures are understood prior to beginning. Blood was considered class characteristic evidence until DNA analysis became available in the early 1980s, which has since sped up identification processes. Because blood is commonly found and DNA can identify individuals, officers should be vigilant in detecting and preserving it. Fresh, uncontaminated blood at a crime scene is easy to identify, but as it ages or mixes with substances like dirt or paint, identification becomes harder. Blood stains can range in color from rust to gray, black, green, or blue. Small drops may require viewing surfaces at an oblique angle for detection. Blood stains at crime scenes form distinct patterns influenced by wound characteristics, weapon type, victim and offender movements, body relocation, and any postmortem violence, which may indicate rage or a prior relationship. Blood stains may be found in numerous locations. One method for locating blood evidence is the use of a presumptive or preliminary field test. Luminol, a water-based spray, detects trace amounts of blood within 30 seconds, producing a blue glow when the lights are turned off. The limitations of luminol include a brief glow duration, inability to differentiate between human and animal blood, and reactivity to copper, certain alloys, and specific bleaches. Blood samples from individuals should only be collected by qualified medical personnel. Laboratory tests on blood evidence can reveal species, for example, human, dog, horse. Gender, blood type and DNA. Drug or alcohol use. Certain illnesses, like venereal disease. Carbon monoxide exposure. Whether a person smokes. Firearm evidence includes revolvers, semi-automatic handguns, rifles, scopes, shotguns, various ammunition types, shell cases, gunshot residues, magazines, and tool marks like firing pin or extractor impressions. Investigators need a solid understanding of firearm evidence because they encounter it often, for safety reasons, and for its importance in gunfight scenarios. Laboratory examinations can help identify whether a specific bullet was fired from a particular weapon. Shotguns are smooth bore, while pistols and rifles have rifled barrels. Caliber refers to bullet diameter, bore is the barrel's inside diameter between lands, and grooves are the low sides. When fired, the slightly larger bullet is gripped by the rifling, causing it to spin, usually rightward, which improves range and accuracy. Information from bullets and cartridge cases include: fired bullets reveal weapon class traits such as number and dimensions of lands and grooves. Cartridge cases show unique marks from firing forces, which may provide individual evidence. Other firearms evidence may reveal: examinations may reveal weapon modifications. For example, was a firearm altered to fire automatically? Sensitive triggers or defects can indicate potential for accidental discharge. Analysis may confirm if a weapon could fire when dropped on its hammer. Serial numbers can often be restored for identification. Tool marks fall into three main types. 1. Impressions from direct force, like a hammer strike. 2. Scrapes from lateral movement, such as a pry bar. And 3. Pinching/shearing by tools like bolt cutters or pliers. Examiners assess tool marks to identify the tool type, evaluate evidentiary value, determine tool motion, describe tool size and features, note unique aspects, e.g., damaged pry bar tip, check if parts were once joined, and see if characteristics match the suspect tool. They may also find evidence to be inconclusive or rule out the tool as the source. Loosely defined, a document is anything on which a mark, symbol, or writing is made for the purpose of transmitting a meaning. A disputed or questioned document is one whose origin or authenticity is in doubt. Handwriting samples can be requested or non-requested. Some labs offer booklets for collecting requested samples. Non-requested writings aren't usually disguised, as they're not meant for examination. Disguised handwriting poses challenges for document examiners and often leads to misleading or inconclusive authorship opinions. One study found a 4.3% error rate in labeling writing as genuine or disguised. Forensic document analysis can verify authenticity, detect forgeries, recover damaged or indented writing, identify writing tools and inks, trace paper sources, detect alterations, estimate age, and link documents to specific office machines. In summary, evidence collection should preserve document integrity. Investigators must avoid altering, repairing, or folding items. Follow these general steps when collecting evidence. Use gloves and clean tweezers to minimize handling. Store documents in a transparent envelope or manila paper envelope. Complete envelope information before sealing the document inside. Leave latent print and DNA processing for the lab. Keep documents cool, dry, and away from sunlight until submission. Have a great day.